Hi everyone, and thanks for coming to our talk on evolution of digital endosymbiosis. I'm Kiara Johnson, an undergraduate student at College College, and I will be presenting work in collaboration with Sylvie Dirkswager and Professor Anya Vostinar. To provide a roadmap for this presentation, I'll first discuss the background for this research. What is endosymbiosis? We'll cover the implications of this research and future work that could be done with the system, and finally walk through the question. First, what is endosymbiosis? Why does it matter? And what makes evolution possible to model digitally? Symbiosis is a sustained relationship between two species over evolutionary time. It can range from beneficial, mutual, to antagonistic, which is parasitism. These relationships can also exist in several spatial dimensions. Endosymbiosis involves one organism, an endosymbiont, living within another, a host, while ectosymbiosis involves the symbiote living on the host. Common examples of endosymbiosis include mitochondrial ancestors, which it's claimed arrived in eukaryotes through endosymbiosis, symbiodinium, which are dinoflagellate endosymbionts of several marine organisms, such as corals and giant clams, and provide a variety of metabolic benefits. And finally, Plasmodium, which is the genus of malaria-causing endosymbionts carried by Anopheles mosquitoes. Meanwhile, some well-known examples of ectosymbiosis include human head lice, which are parasitic, and the group commonly referred to as cleanerfish, who consume ectoparasites and surface-level refuse of their hosts. These examples demonstrate the variety of spatial and behavioral dynamics observed in natural symbioses. Evidently, symbiosis is widespread in nature and is therefore an important dynamic to study. Our research centers around the evolution of endosymbiosis, how we go from individual, separate species to a dynamic where one lives within the other. Why do we study endosymbiotic evolution? As stated, endosymbiosis is thought to have been crucial to the incorporation of mitochondria and eukaryotic ancestors. This endosymbiotic origin of mitochondria then may have provided energy and genetic complexity for the diversification of the eukaryotic clade. In the modern day, many complex multicellular organisms consist of eukaryotic cells. Thus, understanding how endosymbiosis evolves can help us better understand the origin of mitochondria and other endosymbioses crucial to modern complexity. Natural organic evolution has occurred on the timescale of billions of years. It could take years, if not decades, to study evolution in a wet lab. However, we believe that we can model evolution digitally, and thus much more quickly and cheaply by breaking it down to only 4 key components. These are variation, organisms need to have differences that affect their fitness; inheritance, organisms inherit fitness-affecting traits from their parents; competition, organisms who are more fit produce more offspring; and time, many generations occur. Using these 4 basic principles, we can then model evolution under different conditions and in different forms, including modeling the evolution of endosymbiosis. In this presentation, our lab asks the question, how does the possibility of ectosymbiosis influence the evolution of endosymbiosis? Do symbionts who live independently from hosts give up their independence and become endosymbionts at a different rate if it's also possible for those symbionts to interact with nearby hosts while living outside of them? Do the endosymbioses that evolve in the presence of ectosymbiosis behave differently than those who don't? To ask these questions, we use our digital agent-based model of evolution called simulation. Given that digital models of evolution are feasible, the reason we use one in this context is because it provides a number of advantages. The first is control. We are certain that endosymbiosis and ectosymbiosis are both possible in this system because we have written them as evolvable behaviors and all of the parameters affecting the system and their values are known to us. The second is replicability. We see it our random number generators with recorded values so that each experiment can be replicated and the results can be regenerated. Finally, digital evolution is very quick. Tens of thousands of updates can be simulated in a matter of hours. The simulation model consists of 2 grids. One stores hosts and the other stores free-living symbionts. Free-living symbionts do not interact with hosts unless they evolve ectosymbiotic functionality, in which case they can behave symbiotically with the host in the corresponding position in the host grid. Free-living symbiotes can also evolve the ability to infect, at which point they leave the free-living symbiote grid and try to enter the host at the corresponding grid position. If the host already has a predetermined limit number of symbiotes, the infecting symbiote fails and is killed. Otherwise, it succeeds and becomes an endosymbiote. When organisms accrue a certain set number of points, they reproduce. Symbiotes can reproduce in three ways. First, endosymbiotes can vertically transmit. They can inject their offspring into the offspring of their host. Second, endosymbiotes can horizontally transmit. They can inject their offspring into the free-living symbiote world, where the offspring can then try to infect a different host. Thirdly, free-living symbiotes can release their offspring into the free-living world. All three of these modes of reproduction have their own fixed resource costs, which are set at the beginning of the experiment. Sylvie will now explain how digital genomes operate and how organisms gain points.
So the way our organisms, both hosts and symbionts, and their genomes work is very similar to a vita. Organisms have genomes that represent programs, and they're made of individual low-level instructions. And they need to complete logic tasks by building more complex logical operations out of these simpler ones. When they complete tasks, they gain points, and they can spend those points to reproduce using a special instruction for reproduction. The set of instructions and tasks that we use is modeled after a vita's. But unlike a vita, our organisms aren't acting alone. Hosts and symbionts, both ecto and endosymbionts, can interact in three different ways. First, a symbiont can donate or steal resources from its hosts, which is an easy way to exhibit mutualism or parasitism. However, this only applies to ectosymbionts, as mentioned earlier. Next, some low-level tasks can only be completed once every few updates, and this limit is shared between hosts and symbionts. So if one completes a limited task, the other will have to wait until the limit resets, which can lead to parasitism. Finally, some tasks depend on other tasks which need to be completed first, and those completions can also be shared. So if a symbiont completes a task, for example, its host can then complete a different task that depends on the first task. Note that organisms can choose whether their task completions are available for dependencies in this manner, and while symbionts can choose arbitrarily, for hosts, if they choose to prevent their symbionts from accessing their dependencies, this comes with a 20% resource penalty. In this way, symbionts and hosts have many ways to interact and can come up with complex forms of mutualism or parasitism.
What did this model determine about the influence of ectosymbiosis on the evolution of endosymbiosis? We can further break this down into three sub-questions. First, speed. How quickly does endosymbiosis evolve on average in the presence and absence of ectosymbiosis? Second, endosymbiont behavior. Does the presence of ectosymbiosis affect the level of parasitism or mutualism that endosymbionts exhibit? Third, symbiote behavior. When ectosymbiosis is permitted, do endosymbionts and ectosymbionts treat hosts differently? So, first, speed. What we find is that the possibility of ectosymbiosis hastens the evolution of endosymbiosis. In this plot, the x-axis is time and the y-axis is the number of endosymbionts. The colors indicate how many resources are required for endosymbiont horizontal transmission. We observe very rapid increases in endosymbiont count when the resources required for endosymbiont horizontal transmission are lower, the blue lines. When the resources required for endosymbiont horizontal transmission are higher, we can clearly see how in the left-hand plot, where ectosymbiosis is not permitted, the purple line is less steep than in the right-hand plot, where ectosymbiosis is permitted. So, we can conclude that ectosymbiosis hastens the evolution of endosymbiosis in the system, and that endosymbiosis evolves more rapidly when fewer points are required for endosymbiont horizontal transmission. Now on to the second sub-question. Do we observe different levels of parasitism or mutualism when ectosymbiosis is permitted? We find that endosymbionts are more parasitic when ectosymbionts are permitted. In the plot on the screen, the y-axis represents the ratio between points stolen from and points donated to hosts by endosymbionts at the time point at the end of the experiment. The value of 1 means that the amount stolen is equal to the amount donated, so is functionally neutral. This number is represented by the dashed line on the plot. Ratios of less than 1 indicate mutualism, as more points are donated than stolen. Higher point ratios indicate higher levels of parasitism. Colors, again, indicate Colors, again, indicate the number of resources required for endosymbiotic horizontal transmission. The x-axis represents the vertical transmission rate, which is a probability that an endosymbiont will inject its offspring into its host offspring. In past work with our system, higher vertical transmission rates generally produce higher levels of mutualism, as it creates a tighter relationship between single lineages of hosts and symbionts. In this particular experiment, we see that higher vertical transmission rates occasionally produce single experiments that have mutualistic endosymbionts when ectosymbiosis is not permitted. These experiments are outliers. The average end state for all experiments, ectosymbiosis permitted or not, is parasitic. However, endosymbionts are on average more parasitic when ectosymbiosis is permitted. Furthermore, when ectosymbiosis is not permitted, a higher number of resources required for endosymbiotic horizontal transmission translates to lower levels of parasitism. When ectosymbiosis is permitted, the number of resources required for endosymbiotic horizontal transmission does not have significant effects on endosymbiotic behavior. The key points from this section are that the possibility of ectosymbiosis produced more extreme and more consistent parasitism, and that when ectosymbiosis is not permitted, higher endosymbiont horizontal transmission resource costs lead to lower rates of parasitism. The third sub-question asks how ectosymbionts and endosymbionts differ when ectosymbiosis is permitted. This plot has the same axes and color scheme as the last. Here, we see that lower cost of endosymbiotic horizontal transmission produces more parasitic ectosymbionts, imitating endosymbiont behavior trends when ectosymbiosis is not permitted. When ectosymbiosis is permitted, endosymbiont behavior is not particularly affected by endosymbiont horizontal transmission cost. Neither ectosymbionts nor endosymbionts are affected by vertical transmission rate in this condition. Due to the influence of endosymbiont horizontal transmission cost on ectosymbionts but not on endosymbionts, the greatest difference between the behavior of these two populations is generally observed when endosymbiont horizontal transmission cost is low, and the populations have more similar behavior when endosymbiont horizontal transmission cost is high. Key takeaways are that ectosymbionts generally evolve to be more parasitic than endosymbionts in the same system, and ectosymbionts are generally more parasitic when the cost of endosymbiont horizontal transmission is lower, but endosymbionts are not affected by this growth. In our system, we found that endosymbiosis evolved more rapidly when ectosymbiosis was possible, endosymbionts were more parasitic when ectosymbiosis was possible, and ectosymbionts were more parasitic than endosymbionts in the same system. There are several possible explanations for these trends. It could be that ectosymbiosis provided symbionts with extra resources they needed to reproduce, and thus evolve more quickly, leading to more rapid evolution of endosymbiosis and more extreme behavior. Further work that could be conducted to examine the particulars of this system include phylogenetic tracking of digital organisms, enabling us to track the spread and extinction of lineages, closer tracking of transmission rates, and organism reproductive speed, which would influence how rapidly populations could accrue mutations, and further work with the number of resources certain classes of organisms need to reproduce. And that concludes our presentation. Thank you for your time and attention. Does anyone have comments or questions?